Hello, my name is Carole Lunny with St. Michael's Hospital at the University of Toronto and the Cochrane Hypertension Group at the University of British Columbia. I'm based remotely out of Squamish, British Columbia, Canada, and today I'll be talking about the development of a new risk of bias tool for network meta-analysis on behalf of myself and my co-authors. Please tweet us at Carole_Lunny and use the hashtag NMA. So we've received funding for this project through a 2021 successful CIHR project grant, which covers 3 years of funding. We have no conflicts of interest to declare, and we wish to acknowledge the traditional, ancestral, and unceded territory of the Musqueam, Squamish, and Tsleil-Waututh nations on which the research operates in Squamish, British Columbia. Our project team consists of the steering committee, our stakeholders, and the working group. The steering committee consists of myself, Julian Higgins, Ian White, Sophia Diaz, Andrea Trico, Brian Hutton, R.G. Veroniki, Jim Wright, and Penny Wedding. We're also working with students and an international group of stakeholders who will help us in our dissemination efforts. So for background. Network meta-analyses can compare all treatments for a condition, whereas traditional meta-analysis can only compare two. Network meta-analyses provide evidence to clinicians faster than traditional meta-analyses due to the dearth of evidence from head-to-head comparative studies. For example, researchers recently showed that prostaglandins would have been identified 7 years earlier as the most effective drug class in lowering intraocular pressure for open-angle glaucoma if an NMA had been done at that time. So NMAs can help patients and their care providers choose the treatments that's most important to them based on side effects and efficacy of all treatments. So why is it important to assess network meta-analyses for bias using a risk of bias tool? Without a risk of bias tool for network meta-analyses, knowledge users will not be able to know if an NMA is at low risk of bias and whether to trust the results. So multiple network meta-analyses are published on the same topic, and choosing from conflicting NMAs is impossible without a risk of bias tool. So in the study by Metrics authors on the right, the authors found 28 NMAs on the treatment for rheumatoid arthritis, all with differing, conflicting, and discordant results. So if an NMA is at risk of bias and inappropriate methods are used, the validity of the findings can be compromised, which can then impact on patient treatment and care. Conceptual decisions were made about the tool by the steering committee, about the type of tool we want to develop. They were, first of all, we wanted to use a domain-based structure with signaling statements to make the tool easier to understand and more transparent. And the statements are answered with the options yes, probably no, no, or no information. Risk of bias judgments follow from the answers to the signaling statements being low risk, some concerns, or high risk of bias. So our tool is intended to supplement an assessment of the biases in an underlying evidence review, which ideally would be a systematic review. And RoB is currently the only tool designed to assess biases in systematic reviews, and so our tool is designed to complement a RoB assessment. Finally, our tool will be used in NMAs of all study designs, not just randomized control trials. So our definition of a network meta-analysis was a synthesis of evidence on the relative effects of 3 or more interventions simultaneously. And the purpose of our risk of bias tool is to assess the degree to which the review methods lead to a risk of bias in the review conclusions and the results. We aim to provide a tool to assess biases and limitations in the way in which the analysis was planned, analyzed, and presented, including the way in which the evidence was assembled. If these methods were inappropriate, we thought that the validity of the findings would be compromised. So here, this table shows that several tools are designed with different purposes. Some for assessing reporting quality or reporting comprehensiveness, and some for assessing the quality of conduct. But none were designed to assess the risk of bias in network meta-analyses. So for critically appraising the quality of reviews, the AmStar 2 checklist is the most commonly used tool for systematic reviews, and it aims to evaluate the quality of conduct of reviews with pairwise meta-analysis. The RoB is a domain-based tool which is used to assess the risk of bias in systematic reviews, results, and conclusions based on pairwise meta-analysis. To assess the reporting quality of network meta-analyses, users could use the Prisma NMA checklist or the NICE DSU checklist. And then finally, to assess the quality of conduct, users could use the 2014 iSPOR checklist. However, due to important methodological advances in the field of NMA, the iSPOR checklist published in 2014 is currently outdated. So for our purposes, there has been no risk of bias tool for network meta-analyses developed to date, so we are doing that right now. So we'll now move on to the risk of bias in an NMA tool development. Our objectives. So on to the methodological review of items. We included articles that described items used to assess bias in network meta-analysis and searched Medline, Cochrane, methods collections, and gray literature. We extracted characteristics of NMA tools or methods, and the items were initially extracted verbatim. We then determined if the item would be included if it was potentially relevant to bias quality or reporting in NMAs. It was also excluded if the item pertained to a general systematic review method covered by AmStar or RoBus. For the analysis, we grouped items that were similar, split complex items so that each covered a single concept, and we categorized them as related to NMA specific biases and grouped them into domains. Screening and data extraction, 59 studies were included, yielding 14 tools, checklists or standards, 12 guidance documents, 26 articles related to bias or methods, and 7 papers assessing the quality of NMAs. A final list of 22 items were included, which were converted to 22 concepts, which were just basically statements. And they were categorized into 3 domains: network characteristics, effect modifiers, synthesis, and interpretation. And finally, items were entered this past summer into a Delphi survey for experts to get their opinion on whether the items should be included or excluded. So the next study we conducted was a multi-round Delphi survey with network meta-analysis experts to select items to include in the tool. So the Delphi method is a consensus group method used to synthesize expert opinions when evidence is lacking, limited, or contradictory. So we used Qualtrics as a survey tool and got ethics approval through the University of British Columbia. we conducted 2 rounds of surveys, and the steering group suggested experts in NMA and bias. We ended up emailing 50 experts. The survey contained 22 of the items based on and rated on importance using a 5-point Likert scale, and items that were included based on 70% agreement among Delphi respondents. Items comments were discussed by the committee, and items were reworded finally into concepts. Participated in round 1 and 22 in round 2. So here in the table, the first column indicates whether the item was included based on 70% agreement. Blank responses and unable to score responses were not counted in the denominator. And finally, 7 out of 22 concepts did not reach agreement in round 2. We conducted a survey to ask decision makers about the structure and development of the tool. Again, for this survey design, we used Qualtrics and got ethics approval through UBC. The survey ran this summer. We tried to recruit any type of decision maker and institution producing NMAs. Our recruitment was through emails of participants through UBC Method Speaker Series. We also advertised through social media. We invited anybody basically to take our survey, and we included 12 closed and 10 open-ended questions which were anonymous. The average time to take the survey was 2 minutes, and denominators were taken as the number who provided a response to the question. So the results of the survey were that 2,734 emails were sent out, and then 297 or 14% were complete and were included in our analysis. A lot of the respondents had several roles that they identified with, mostly being systematic review or academic. And then third most prevalent was clinician or allied healthcare provider or guideline developer. About 10% were journal editors. And about 75% said that their organization or institution produced systematic reviews with network meta-analysis. Again, 65% said that they used network meta-analysis as a source of evidence in decision-making, and the majority said that they would like a tool to analyze both individual results and conclusions in network meta-analyses. So now we'll just take a little preliminary look at the structure of the risk of bias NMA tool. Here is a preliminary look at the structure of the tool. However, the content has not been finalized, so I can't present the questions or the statements today. So there's 3 domains to the tool, seen here on the left: network characteristics, effect modifiers, and synthesis. So domain 1 contains 4 signaling statements, domain 2 contains 3 signaling statements in yellow, and domain 3 in blue has 7 signaling statements. After answering the signaling statements in each domain, the assessor then answers 2 overall judgment statements, seen here in green, and finally makes an overall judgment of low, high, or unclear risk. In conclusion, rigorously developed methodological and reporting tools for NMAs published from 2012 to 2014 are now outdated and fail to incorporate methodological advances in NMA conduct. So our new tool will be current and aims to incorporate new advances. Our methodological review of item yielded 22 concepts. Our Delphi and Decision Maker survey were conducted this past summer to further refine the items in the tool. And these studies provided the groundwork for the development of a new tool to assess the bias in network meta-analysis, which will provide a way to choose NMAs that are at the lowest risk of bias. Our future work will be to do pilot testing, and if you're interested, please get in touch with me to be part of that. I thank you for your attention, and I welcome any questions that you may have. Thank you.